And we are live. Welcome everyone to the 5th installment in our webinar series. I'm just gonna start this off real quick. Technical things. Here we are. All right, there we go. Welcome everyone to the 4th installment in our freshly rebranded webinar series, now called Swedish Research in Cell Biology. You are all here to listen to a lecture from Oscar Bedoya Reina at KI, and his lecture today is going to be titled Single Nuclei Transcriptomes from Human Adrenal Gland Reveal Distinct Cellular Identities of Low and High Risk Neuroblastoma Tumors. And this is an event hosted by us at Milmed here in Sweden, and together with our good friends over at Baker Ruskin. Sadly, Krista Rantanen from Baker Ruskin had to cancel today, so I'm going to present both my part and then also Baker Ruskin's part. So before we get into today's main event, I'm just going to give a little brief presentation of this lecture and my brand and company, as well as Baker Ruskin. So today's agenda, I will present Milmed, Baker Ruskin. You get to listen to Oscar, and then we're going to have a live Q&A session. After that, I'm also going to announce our next webinar, which is in December. I realized that I've written that incorrectly there. But yeah, so next webinar in December. I might switch the last 2 points depending on the number of attendees we have. So yeah, depending on the number of attendees, if we're within the limits of this program or not, we will either stay here or move over to Zoom. So we'll discuss that when we reach that point. The idea is that you get to discuss your questions and ideas live with Oscar instead of just writing them down. All right, so Milmed, we are a Swedish company located in the south of Sweden, and our slogan is Tailored Laboratory and Industry Technology. We do everything from supply, install, and maintain simple laboratory equipment to advanced laboratory equipment and also industrial equipment. And we're a general agent in Sweden to several leading brands worldwide, including then, of course, Baker Ruskin, but also brands such as Coyer, Fedegari when it comes to sterilization, Etaluma microscopes, and many, many more. Today we're going to talk about neuroblastoma. So what conditions would be optimal for working with neuroblastomal cells? Well, here at Milmed, we are firm believers of that physiologically relevant results arise from physiologically relevant culturing conditions. And in order to acquire such conditions, we recommend, for example, in vivo 400 by Baker Ruskin, where you create or simulate a in vivo environment for your cultured cells. You control parameters such as temperature, humidity, carbon dioxide, and of course then also oxygen. They're also provided with internal HEPA filtration as standard. You can read more at our website, milmedtech.se. If you're in Sweden, it's in Swedish. Or of course, if you're outside of Sweden, you can always visit Baker Ruskin's website as well, written down at the bottom of the screen here. Oxygen and neuroblastomas, well, you could go into a lot of details there, and we don't have that time, but some suggested reading for you is by our good friend Sophie and her colleague Sven Pohlman. A good review of hypoxic conditions in neuroblastoma. So some recommended reading where you can find some more. Now then, since Krista from Baker Ruskin is unavailable, I'm going to present some of her slides as well from Baker Ruskin's perspective. I'm just going to select the correct presentation. There we go. So Baker Ruskin provide hypoxia or physoxia workstations But they're a part of a great organization called Baker. They're also there together with Clean Air. You might have heard of these brands before. Pioneering brands, all of them. Baker were the first with introducing the first biological safety cabinets in 1948. Baker Ruskin pioneered the first hypoxia workstation in 1999 as well. And Clean Air has been working with biological safety cabinets since the 70s. Baker Ruskin would like to formally invite you to Hypoxia U, which is a virtual event series where they have lectures regarding hypoxia. So it's quarterly online seminars designed to encourage networking and communication of novel concepts. Short lectures and poster sessions. It's going to be a lot of unpublished work on the biological responses to variations in oxygenation. Hopefully, there will be a live meeting as well. Pandemic pending, of course, but keep a lookout for that in 2022. Hypoxia U has a well-known scientific organization committee where we recognize names such as Roland Wenger, Randall Johnson, and Chris Pugh, among others. And Krista, who couldn't be here today, will be the moderator of Hypoxia U. Save the date of the next event, which is December the 9th. And you can find more information about that on hypoxiau.com. Krista, you can follow Krista on Twitter under the name Dr. Ox, where she regularly posts science related to hypoxia in various types of ways. She also has a YouTube channel associated with that, where you have the series called Unexpected Questions, which interviews leading researchers worldwide. And then, of course, she asks them unexpected questions. All right. So if you're in the need of microbiological safety cabinets or hypoxic equipment, feel free to contact us or Baker Ruskin and we'll set you up. With that short introduction, I'm going to have the privilege then to present our today's lecturer, Oscar Bedoya Reina from KI. So please, Oscar, join when you're available.
Hello.
Let's see. Here we go. And I'm going to hide as well. So that'll be in the background.
Great. I want to thank the organizers of this seminar for inviting me to give this talk. My name is Oscar Bedoya, and I am going to present the results of my research. These results were recently projects, and this presentation has for title Transcriptional Differences Between Low and High-Risk Neuroblastomas from a Single-Cell Perspective. Neuroblastoma is responsible for 10% of the cancers in pediatric children, while it is responsible for 15% of these deaths because of cancer. Danny, I am hearing myself, like feedback is kind of distracting. Can you fix it out?
That feedback, I believe, is probably something local on your computer. I don't think I can do that. What you could do is turn off just your headset if it's distracting.
I can do that. Okay. As I was saying, neuroblastoma is responsible for 10% of the cancer cases in children, but it is responsible for 15% of deaths in pediatric because of pediatric oncology. It is the case that neuroblastoma has large clinical heterogeneity as a hallmark and ranges from favorable localized differentiated tumors to favorable metastatic and aggressive tumors. And there is, interestingly enough, a stage that is arguably metastatic that spontaneously regresses without further treatment. It is the case that neuroblastoma comes in two different forms. One, tumors, a set of tumors that have favorable outcome and low risk, and a set of tumors that have high risk and unfavorable outcome. And one of the main factors that define what is a low risk and a high risk neuroblastoma is the age at the time of diagnostic. And this threshold is 18 months. So children younger than 18 months are generally diagnosed with favorable neuroblastomas, low risk, and children older than 18 months are commonly diagnosed with unfavorable neuroblastomas. Neuroblastoma is a malignancy of the sympathoadrenal system and thus have neural crest origin. It commonly occurs in the adrenal medulla, and it is thought to originate during development as it is a pediatric cancer. A large body of evidence from the development in the sympathoadrenal system comes from the work of recent years from Igor Adameiko's group. In the first model that you can see on the left side of your screen, what they proposed in their 2017 science paper was that there were two different cell populations or migrations that populated the adrenal medulla. The first was an early migrating population of cells from the neural crest that at the end ended up originating about 20% of the cells in the adrenal medulla. And later, there was a second wave of cells migrating to the nerves that originated about 80% of the cells. Those cells that migrate through the nerves are Schwann cell precursors, and their original model was set up here. So originally, they suggested that Schwann cell precursors transiented into chromaffin cells to originate them, but they didn't find a connection to sympathoblast. This model was revisited in recent months with more data, and what they concluded was that the Schwann cell precursors originate chromaffin cells, and further they originate sympathoblast. Nevertheless, it is the case that this is the model for mouse. And of course, the question is how similar is mouse during development to human? And we know for sure that at least one gene that is important in the sympathoblast development in mouse is otherwise expressed in chromaffin cells in humans. Additionally, in the same paper from Igor's group this year, they suggested that in humans, it is possible that Schwann cell precursors originate both chromaffin cells and sympathoblasts as opposed to humans. Then the next question is, of course, how chromaffin cells, this is medullary cells in the adrenal gland, are repopulated postnatally. We understand from the body of evidence that Lopez group has collected over the years that it is possible that the chromaffin cells in the karyotic body originate from either a neuroblast or glial sustentacular cells, and this depends upon the intensity or the time of exposure to hypoxia. Then, of course, it is the case that we wonder how these populations in development adrenal gland and in postnatal adrenal gland are similar to low and high risk neuroblastomas. So keeping that in mind, we wanted to explore the similarities. And to do so, what we did was comparing neuroblastoma sequences from a single cell perspective and compare it with developing and postnatal human adrenal gland as well as mouse postnatal and developing adrenal gland. We were interested in answering questions, the following questions. How similar are cells in human and mouse adrenal? How similar are cells in developing and postnatal adrenal? and how cells in neuroblastoma changes with risk and age. To this end, as I mentioned, we sequenced 3 postnatal adrenal glands in humans, 5 from mouse, and 11 neuroblastomas, including 5 low-risk tumors and 5 high-risk tumors. We use a strategy that allows us to obtain high-quality data at the cost of a lower number of nuclei or cell sequence. In particular, when processing the mouse and human samples, we used somehow different methods. Mouse samples come from fresh samples, so the cells that we obtain are fresh cells and included RNA from cytoplasm. And oppositely, in humans, we obtain deep frozen samples, and we sorted the frozen, processed the frozen nuclei and sorted the RNA from those nuclei. So after sorting these cells and nuclei RNA, we sequence them using a SmartSeq2 technology. We process this data using a strong quality controls for reads and for cells, and further process this data with the Pagoda pipeline. It is important to keep in mind that human data and mouse data have different features associated with their origin, meaning that human data presents characteristics of RNA coming from nuclei, including a larger number of reads mapped to intragenic regions and a larger number of reads mapped to intronic reads. And oppositely, mouse have a larger number of reads mapped to exonic regions and to intragenic regions as well. We cluster the cells from both human adrenal glands on the left and mouse adrenal glands on the right, and we found commonalities and differences. In particular, we could identify, as expected, clusters of cortex in human in here and in mouse in here, and a number of other clusters, including immune clusters, macrophages and T cells, mesenchymal cells, and endothelial cells in both adrenal glands. Nevertheless, it was the case that we identify a population of glands, a population of cells, sorry, in the human adrenal gland here in pink, and that population of cell express progenitor markers as indicated here. Oppositely, in mouse, we identify 2 different clusters of chromatin cells in here, while in human, we just identify a single cluster. These 2 clusters of chromatin cells in mouse expresses a different set of muscarinic and nicotinic acetylcholine receptors, as you can see in this part. But perhaps more important, they also have a different expression of PNMT. PNMT catalyzes the transformation from noradrenaline to adrenaline, which is one of the most important endocrine functions of the adrenal gland in the fight or flight response. We tested whether one population of the chromatin cells could originate the other population of chromatin cells, and found no evidence that it was the case. Rather, the velocity analysis in here suggests that the differentiations of the chromatin cells in mouse takes a different path in this direction and another different path in this other direction. In regards of the population of cells in the human adrenal gland that express these progenitor markers, including ARBB3 and RTTN, as I indicated before, we found 3 additional computational evidence that it is the case that these cells source or originate chromatin cells or turn into chromatin cells. The first one comes from the velocity analysis, in which, as you can see, the cells progenitors convert or direct towards the more differentiated chromaffin cells. And similarly, the differentiation potential mentioned as entropy is higher in the progenitor cells as compared to the chromaffin cells. And the progenitor cells are located early in a pseudo-time trajectory as compared to the chromaffin cells. In this pseudo-time trajectory that you can find on the right side, these progenitor cells start expressing mesenchymal and motility markers and end up expressing more differentiation and adrenergic markers, including TH and PNMT. Further, what we tried to do was locating these populations of cells in the adrenal gland in human, and we found two interesting things. The first one is that this population, so what you can see here is 0-year-old adrenal gland on the left for an adult adrenal gland, and this is also 4 years old adrenal gland. And what we could see is that this population, using RNA-scope, was present in the capsule of the adrenal medulla and in the of the adrenal gland and in the medulla itself. Let me try to explain this a little bit better. So this part is the capsule of the adrenal gland. This part is the cortex, and this part inside is the medulla. So this population is present in both the capsule and in the medulla, and the population seems to decrease with age, being higher in just borns and decreasing with time. Next, we computed the significance of the genes shared between these clusters, and we could see that the chromaffin cells in humans and in mouse share a significant number of genes, as well as macrophages, endothelial, and mesenchymal cells. We move then to characterize the neuroblastoma cells. We obtained 10 different clusters, and these clusters could be broadly grouped into a noradrenergic cluster in blue color, stroma clusters here in the bottom that include a mesenchymal stroma cluster and an endothelial cluster, immune cells here, and two clusters of undifferentiated cells in here. Interestingly enough, the NOR clusters express, as expected, high levels of noradrenergic markers, while the undifferentiated cluster poorly express noradrenergic markers and rather express the unfavorable markers that we have here, including NTRK2, MECAN, BRCA1, and ALK. This is another way of visualizing these results. The undifferentiated population presented unfavorable markers, some mesenchymal markers and progenitor markers, and the more noradrenergic clusters significantly express noradrenergic markers. It has been reported in the literature, and we suspected as well from our data that lower risk neuroblastomas have a more differentiated nature, features, and they express differentiation markers, adrenergic differentiation markers, while the undifferentiated clusters are rather associated with high risk and do not express those adrenergic markers so strongly. So we look then at the origin of the cells by a patient, and we did see that the clusters, the NOR clusters, were more likely to come from low-risk patients, while both the mesenchymal and endothelial stroma and the undifferentiated clusters were more likely to come from high-risk patients. We properly tested this hypothesis, and we could see as we suspected, that the mesenchymal stroma and endothelial clusters, and both undifferentiated clusters, presented a significantly higher proportion in high-risk neuroblastoma, while the noradrenergic clusters 5, 7, 8, and 9, all of this over here, were significantly representing a significantly higher proportion in low-risk neuroblastomas. One caveat of our study is, of course, that we are using 11 neuroblastomas for trying to characterize a very heterogenic malignancy. To try to bridge this approach, what we did was using a computational approach to try to see the proportion of these populations in 172 neuroblastoma sequence tumors, previously sequenced by the target group. Then what we could identify was that in those tumors, 172 tumors, we could recapitulate that there was a significant enrichment for cells in the mesenchymal stroma, in undifferentiated cells, the cluster number 3, and endothelial cells, while there was an enrichment for cells in the NOR7 cluster, NOR8 cluster, and NOR9 cluster in the low risk. These were consistent between the pulse sequences and our 11 neuroblastomas, but nevertheless, there were some difference, for instance, in the undifferentiated cluster 2 and in the noradrenergic cluster 5. What we tried to do was to assess then if this undifferentiated NC3 cluster presented the genomic rearrangements that has been characterized for the worst outcome malignancies, in particular 1p loss, 11q loss, or 17q gain. We on top see the computational prediction, and what we can see is that here are the patients in the bottom. For most of the patients, we could see that the undifferentiated cluster NC3 did have a significant rearrangement as expected, while for some cases, the NOR cluster 5, the undifferentiated cluster 2, and in one patient, the mesenchymal stroma cluster also presented this sort of arrangements. We further confirm that it was the case using a SNP microarrays, and what you can see in the bottom are the cells in those patients, coming from those patients that were confirmed had such rearrangements, suggesting that indeed the undifferentiated clusters have the malignant genomic rearrangement. We then try to see if these markers could be recapitulated in the tumors itself. And in some cases, we see that it is the case that that tumors present an homogeneous expression of the cells of the genes characterized by the single cell analysis. In particular, for this tumor that is a 4S, a low risk tumor, we could see that there was expression of the low risk marker NTRK1 and of the adrenergic marker TH, and it was more or less homogeneously through all the tumor, while it didn't have expression of the undifferentiated marker nor of from mesenchymal marker in almost any place in reality. Nevertheless, it was not the case for all tumors. This tumor that I am showing here is an stage 4 tumor. And what we could see is that actually this tumor could be kind of divided into two sections, the one on top and the one in the middle. The section on top has the expression of MECAN and TH, while the bottom has no expression of TH, but has rather expression of MECAN and ALK genes, both of them high-risk markers. Next, what we tried to do was to characterize if the clusters resemble or had rather adrenergic or mesenchymal transcriptional profile. This is relevant because with these two seminal papers from the Van Groningen group and the Oliver Selate group in 2017, they could characterize that neuroblastoma cell cultures comes in two different flavors, epigenetic flavors, one with a more adrenergic nature and another one with a more mesenchymal nature. It is thought that the adrenergic signature is rather associated with a more favorable lower risk, while the mesenchymal signature is associated with relapse and drug resistance, resistant to treatment. So we tried to see how our clusters in the 11 neuroblastomas recapitulated the signatures for adrenergic cells and for mesenchymal cells, as reported by Van Groningen and Boeva. We could see consistently that the noradrenergic clusters had a higher adrenergic signature as opposed to a mesenchymal signature, while the undifferentiated cluster 3 had a mesenchymal signature and not an adrenergic signature. Furthermore, we had the opportunity of comparing the transcriptional profile of these clusters with the recently published at that time analysis of tumors by Sam Behati group publishing this in this paper of Kildisuti early this year. What we could see is that our tumor clusters resemble more likely their tumor clusters as well, with the particularity that our undifferentiated cluster 3 resemble with a high likelihood only their tumor cluster 3. Their tumor cluster 3 is the one among the 3 clusters that they characterize, is the cluster that is the less differentiated cluster, and in consequence is as well the one that is associated with a higher risk. Of course, then the next question was, what does it imply for the survival of the patients to have some type of cell, undifferentiated or noradrenergic or stroma? So what we did in this case was comparing the expression profile of the different clusters with a larger cohort of 498 patients previously reported but by SECSI. And what we did was then trying to see how genes that were significantly and uniquely significantly upregulated in each of these clusters were associated with survival of patients in this larger cohort. We could identify that genes that characterizes the noradrenergic clusters 7 and 8 have a higher expression in patients with a higher likelihood or survival, event-free survival. Oppositely, the high expression of genes characterizing the undifferentiated cluster 3 were more likely to be present in patients with a lower survival probability, as opposed to patients that had a low expression of the genes characterizing the undifferentiated cluster 3. Then we try to dissect the genes that were associated with risk, age, and survival. I will elaborate briefly upon that, and if I have more time, we'll continue on this. But then what we did was defining the gene sets that were significantly different between high and non-high risk, that were inversely correlated with age and directly correlated with age. This is present in younger patients as opposed to elder patients, and that were significantly associated with better and with poor prognosis. What we could see is that the different clusters have a representation of these different groups of genes. In some cases, it is stronger than in others, but this case analysis was very interesting for the clusters 9 and 3, as both of them presented as genes that are associated with poor survival and better survival, and also that were present in elder patients and in young patients over here, sorry. When we look into how similar were these two groups of genes, those associated with age and with worse outcome, we were shocked by discovering that the set of genes that are associated with poor survival are also significantly correlated with age. And it is never the case that poor survival gene set is inversely correlated with age. So one interpretation of these results is that these two populations of cells have a decision fate to make. They can become in elder patients associated with poor survival, or they can decide to express another set of genes in younger patients that is associated with better survival. We try to look in detail into what were those types of genes in association with survival and age. And what we saw was that for both of these clusters, the better survival was associated with cell adhesion and differentiation, and oppositely, the poor survival was associated in the case of the NOR-NC9 cluster with spliceosome deficiencies and translation deficiencies, and in the case of the undifferentiated cluster NC3 with DNA repair and cell cycle and double strand break. We look similarly to those set of genes associated with age in these clusters, and what we could see is once again that those associated with younger patients in here and in here are more likely to be seen associated with cell adhesion and differentiation, and oppositely, those seen in the noradrenergic cluster 9 are also similarly associated with spliceosome deficiencies, while interestingly enough, in the undifferentiated cluster 3, those genes that are present in elder patients have to do with cell motility that can be understood as metastasis. To finalize our study, what we did was comparing the neuroblastoma clusters with the postnatal human and mouse adrenal glands, as well as with the developing glands. We did this using different approaches. In the first one, we tried to see the significance of the genes that were expressed in each of the clusters. And we could see broadly that the noradrenergic clusters express share a significant number of genes with the chromaffin cells in both mice and humans, while the undifferentiated NC3 cluster share a significant number of genes with the progenitor population in the postnatal adrenal gland, and a significant, less significant, but significant with the mesenchymal cells. Looking into the developing adrenal gland, similarly, these NOR clusters show a significant number of genes shared with chromaffin cells and sympathoblast cells for both mice and human during development. And interesting enough, the undifferentiated cluster 3 also shared a significant number of genes with cycling chromaffin and sympathoblast. Nevertheless, when we look in detail into those genes, we could see that these genes were rather cell cycling associated genes as opposed to genes characterizing chromaffin or sympathoblast cells, like classic genes. In the second approach, what we did was computing a signature score for every single cell. And what this signature score does is indicating how similar is the expression of a reference group of genes in every cell as opposed to a random set of genes. So the higher the score, the highest the similarity of one cell to a reference cluster of cells. And what we saw was that we could recapitulate our results with the first approach, in the sense that the undifferentiated cluster look more alike the progenitor cells in the postnatal adrenal glands, and the NOR clusters resemble more the chromaffin cells. For development, we also saw that the noradrenergic clusters resemble more sympathoblast and slightly less the chromaffin cells for both human and mouse. In the third approach, what we did was running this joint analysis. In this particular case, what it does is choosing among a reference set, in this case the adrenal gland, what is the cluster of cells that most likely resembles the transcriptional profile of every single cell. So what we observe is that cells of the progenitor population, of the undifferentiated population, most likely resemble the progenitor population in postnatal adrenal gland, and that the noradrenergic clusters most likely resemble the chromaffin cells postnatally. In the fourth approach, what we did was calculating the similarities in the full transcriptional profile between the different cell populations of developing mouse and postnatal human and mouse adrenal glands, as well as neuroblastoma. This was done using an Euclidean distance, and here displayed using a TSNE. What we could identify was a cluster of cells, glial cells, clusters of endothelial cells, mesenchymal cells, and for the interest of the talk, we could identify that the NOR clusters in neuroblastoma were more similar to the chromaffin cells of human and to the sympathoblast of both mouse, developing mouse, and postnatal mouse as well. While the undifferentiated clusters look more alike the progenitor cells in human. In conclusion, the postnatal human and mouse adrenal glands presents commonalities and differences. We could see that mouse adrenal glands presented two different types of chromaffin cells, while the postnatal human adrenal gland presented a population of cells with progenitor features. That neuroblastoma clusters displayed undifferentiated noradrenergic and stroma profiles. That the noradrenergic cells, as expected, characterize low-risk neuroblastomas, while the undifferentiated ones characterize high-risk neuroblastomas, and that the noradrenergic cluster is more likely representing an adrenergic signature, while the undifferentiated NC3 clusters more likely a mesenchymal signature. The gene expression in the cell populations is associated to different outcomes. Higher expression of genes in the noradrenergic cells is observed in patients with better survival and opposed in the undifferentiated cluster 3 with worse survival. Expression of genes in the noradrenergic clusters is more likely in younger patients with better survival, and in the undifferentiated NC3 clusters is more likely in correlation with age and worse survival. And finally, that neuroblastoma and adrenal cell clusters share transcriptional profiles. Neuroblastoma and developing adrenal gland share transcriptional profiles, as I said, as follows. The neuroblastoma node clusters resemble the adrenal chromatin clusters postnatally and during development to both the sympatoblast and chromatin clusters, and the neuroblastoma undifferentiated NC3 cluster resemble the adrenal progenitor cluster. I want to thank everybody who was involved in the project. Wen Yuli was the person who processed the samples and who did the in situ analysis with RNAscope. Maria conducted the rearrangements, the rearrangement study. Monica and Petra contributed with the in situ staining study as well, and Susanne led the project. Especially I want to thank also Katerina Larsson and Christopher Julin, as well as Per Kogner, because they provided samples from postnatal human adrenal gland and neuroblastomas. And without them, this study wouldn't have been possible. I also want to thank you for your attention. And I want to say that I am starting my research group, so I am interested in starting new collaborations. My main interest is looking into the evolution from primary tumors to metastasis, and also in scrutinizing mouse as a valid model for human malignancies. Saying that, I thank you you all for your time, and it's time for questions. Thank you very much.
Yes, and thank you so much for a fascinating lecture, Oscar, and for taking your time to present your research here in Swedish Research in Cell. We're very happy to have you. So what I'm going to do for everyone's information now is that I'm going to turn off the